This video is going to be just about skeletal muscle anatomy. So here we're looking at a section of a muscle cell or fiber. There are multiple nuclei in skeletal muscles, and they're right next to the plasma membrane. So they're pushed all the way to the outside edges. Here we're sort of seeing three nuclei along that plasma membrane. And so they're pushed aside by these long organelles called myofibrils. Again, we're seeing this myo prefix referring to muscles. So these are long organelles that are going to be sort of the contractile units for our muscles. They fill up most of the cytoplasm of muscle cells as these myofibrils. And so if we zoom into one of those myofibrils, we have what we're looking at here. And you see these alternating light and dark bands along the length of the myofibrils. And that gives it the striated appearance. You have light, dark, light, dark, light, dark, gives this banding pattern that we refer to as the striated appearance. Light and dark, we call them I and A bands. Think second letter of each of those words. I is the second letter of light, A is the second letter of dark. So that's giving it the light and dark appearance. And a sarcomere is going to be a whole contractile unit here from one Z disc to another Z disc. So let's look at what we are showing in this. I'm going to X out of my picture here. So let's look at what we're looking at here. Okay. So the I band is this light section here. In our sarcomere, we have thin and thick filaments. And so the I band has only thin filaments in it. And because they're only thin filaments, this produces a lighter color that we're seeing. And so the I band is light. The A band includes both the thick filaments here, as well as some portion of it overlaps with some thin filaments. So we have a lot more protein in the A band that we're seeing, and so this appears darker. So that dark A band has thick filaments and some thin filaments. The A band has only thin filaments. They both have middle areas. So the middle of the I band is the Z disc, and a sarcomere generally occurs from one Z disc to the next Z disc. The middle of the A band is called the H zone. It will be slightly lighter in the center here because the thin filaments do not go into the H zone. They can move sort of along the length of the A band, but they do not enter the H zone. So it is just a slightly lighter section in the dark A band. The M line is in the center of the H zone, so that is holding the thick filaments together of our thick filaments. And so we have these units of light and dark between Z discs that create a sarcomere. And these are the tiny contractile units that make up the myofibrils, and they lay end to end to end. And so we have all of these repeating sarcomeres down the length of a single myofibril. And it is the arrangement of these small filaments, the dark and the light, that produce the thin and the thick, that produces the dark and the light banding patterns that we see. So let's zoom in on the thick filaments. These thick filaments are these dark red here that produce our A band. Thick filaments are made of myosin, one of our proteins. They also contain ATP-ACE enzymes, right? So enzymes end in ACE. And in this case, they work on ATP. They specifically split ATP, right? ATP is our energy currency, and we need to use energy to contract our muscles. And so we split ATP, releasing its power. That's what those ATP-ACE enzymes will do for us. Okay, so we have enzymes here. They're going to allow us to use ATP. The middle parts in the H band are smooth, and the ends are studded with these myosin heads that come off of the filaments here, and we'll talk a little bit more about those. Those are going to allow us to contract our muscles. The thin filaments, which we're showing here, those are made of actin. So our thick ones are made of myosin, our thin ones are made of actin. So we have these actin and myosin proteins working together to contract muscles. These are anchored to the Z disc, and they slide towards each other during contraction. This thin filament and this thin filament will slide towards each other, towards the M line during contraction. Again, these will help form our light bands, those I bands as well. So light bands, notice that light bands include, so this is a whole sarcomere. A whole sarcomere includes one whole section of an A band, but like half of a section on either side of an I band. Thin filaments do not extend all the way into the center of the sarcomere. They do leave that H region where the M line is, that is slightly lighter in that smooth region there. A little bit more just generally about the muscle cells themselves. They have a sarcoplasmic reticulum. So again, here we are looking at a section of a single cell or a muscle fiber, nucleus here on the outside. Here's a myofibril with our thin and thick filaments that we were just talking about. They also have a sarcoplasmic reticulum, which is this blue section here. You can see it sort of wrapping itself around most of the cells. This is a special type of smooth endoplasmic reticulum where we have these interconnected tubules and sacs that surround each myofibril. This allows a storage of calcium, which is required for muscle contraction, and it releases this on demand as the muscle fiber is stimulated for contraction. So it will hold that calcium and then release it as needed. And that is the basic anatomy of skeletal muscles.